For people in my age demographic, there used to be a chap on late night TV on a Saturday nights called Hal Todd, and he used to sell stuff that you got steak knives with, but he also had an expression halfway through the B-grade movie that was on at midnight with the best yet to come. So the best is still to come, and starting that is Professor Richmond, Peter Richmond, who's a paediatric immunologist, who's going to talk to us about RSV. Thanks, Peter.
Okay, thanks for that, Lou, and thanks for the invitation to speak at this meeting, and it's great to be back in person again. I have to convince my wife that it is better to have a meeting in person than over Zoom. She say, why? But so RSV, winter's not quite here yet, although coming to Melbourne it feels a bit like it from Perth, I'd have to say. And Terry sort of gave an indication that we've probably never had more results from RSV, major RSV studies released in 12 months. So I've been fortunate enough to be involved in some of those, and hopefully I'll give you a sense of where I think we're at and perhaps where we can go, where we'll be going. So I'd just like to start with an acknowledgement of country. We're obviously the traditional owners of the lands that we all come from and where we're meeting today, and pay my respects to their elders past, present and emerging leaders, and thank them for the wisdom that they provide us, and also remind us that we need to address the inequities that they suffer. Here's my declarations of interest. I have been involved in a number of advisory boards and clinical trials with most of the companies here in this room, and have also received some research support. So this is what I will be talking about today. Now, normally I'd start talking about RSV burden and epidemiology, but we're going to get an excellent talk from Gemma following that, so I'm deliberately not starting that. But for the adult physicians in the room, RSV in children is a major cause of pneumonia and hospitalisation, particularly in the first 6 months of life, although it affects children up to age 5 in particular, with a variety of presentations. And then I'll really talk about how we've got to where we're at in 2022 very briefly, update you on the only prevention strategy that has been recently approved in Europe, nirsevimab, and then talk about progress with maternal RSV vaccination, and Terry touched on that today. Talk a little bit about RSV vaccine for older adults, of which there are now a number of studies that have been released, but unfortunately one of the difficulties is when there's so much data coming out, none of it's actually been published yet. So I have had to borrow from a few press releases and the ACIP meeting, which fortunately publishes all their materials that they use, which I think is a great asset for the whole community. And then finally just mention what's happening within paediatric RSV vaccines. So in the early 50s, RSV was first identified in infants by Bob Chanak lab in the NIH, but shortly after that they applied the same approach to polio by inactivating it, and not only did it not protect against the orphans who were being tested on, I'm not sure about the ethics of that at the time, but they actually got more severe disease when they got RSV, including a number of deaths, and that really had a big impact on RSV vaccine development. And when I came back to Australia in year 2000, one of the first studies I was doing was a live attenuated RSV vaccine that had been made by Wyeth. And although there were some promising early results, essentially it couldn't be made immunogenic enough and not be reactogenic in very young infants who you would target. So we've had to look at other alternative ways of protecting the young infants who are most at risk for disease. But I think one of the major breakthroughs that occurred in around 2013 was that Barney Graham and colleagues at NIH actually were able to understand that the fusion protein changed its conformation when it bound to the cell, and actually if you wanted to measure antibodies you really had to measure the protective neutralising antibodies against the pre-fusion protein, which was the form that's expressed on the virus before it infects the cells, obviously, which is when antibodies are going to have their biggest impact. And that really meant that there had to be a change in the design, and including some companies like GSK that actually had to completely redesigned the vaccine they thought they had designed as a pre-fusion vaccine. So that was a really important factor. And that led to this explosion in approaches to RSV vaccines, and also develops in monoclonal antibodies that I'll also mention, that meant that we could design higher efficacy monoclonals that would last for longer. And in fact, this was happening before the COVID pandemic. So you can see here the different modalities of vaccines that were being used, including mRNA vaccines, were in development prior to COVID occurring. And I think this has not really been talked a lot, but I think that really actually helped accelerate the development of the COVID vaccines very quickly, particularly around stabilising the spike protein. And so that was one of the reasons that we were able to go so quickly. And also the adenoviral vectors were in advanced trials in infants, toddlers and adults at that time. So I think there is RSV certainly had a role in helping us start the pandemic. And this is a snapshot that's taken from the Perth website, shown there, and they update it very regularly. So this is from the start of January, and you can see that they update the changes. And so this is the nirsevimab that has now been approved by the EMA, and this is GSK's maternal vaccine that is no longer being progressed as a result of some safety concerns during the efficacy study. And then you can see which ones have made it into phase 2 or have gone from preclinical to phase 1. So it's a really useful snapshot that you're able to use to see where the different vaccines is. And obviously I'm not going to cover this in 20 minutes. So I'm going to talk about the monoclonal antibodies first, because I think this is the one that we will see first for children. And there's two ways of protecting very young children before they've been vaccinated. Obviously, maternal vaccination got a major fillip from the UK when they introduced pertussis vaccination so quickly and getting such high uptake and demonstrating its usefulness against severe disease early in life. And this is exactly what we need for RSV. We have had a monoclonal antibody, palivizumab, for 20 years, more than 25 years, but its problems were it was not sufficiently effective, it was very expensive, and you had to give at least 5 injections. And we did a study a couple of years ago that showed only about 25% of children who were being given palivizumab actually got their 5 doses that they were recommended. The other advantage of using monoclonal antibodies is that you can be much more flexible about when you provide the protection. So you can also protect older children, which is not necessarily possible with maternal vaccination, although we will find a bit more about that. And so this is a schematic of the differences between the pre and the post-fusion vaccines, looking at the different epitopes that have been mapped. Obviously, one of the downsides with monoclonal antibodies is if you have a significant mutation, as we've heard for COVID, it can have a big impact on efficacy. And in fact, subtavumab was an example of that, where a mutation in RSV meant that it was no longer effective in their phase 3 trials, and so it had to be dropped. The other issue that's important to note is that, as with vaccines, if you have epitopes that are predominantly on the post-fusion protein, they don't work as well, and you can get a sort of a scale of going from which ones have the highest sensitivity for neutralisation of antibodies compared to those that have the lowest, where you have more antibody directed against the post-fusion epitope. So this is important, but is not necessarily the whole story. Now, nirsevimab was developed by AstraZeneca or MedImmune, who had produced palivizumab, and they were able to demonstrate that particular epitope had much higher neutralisation antibodies, as you can see in this slide. And they also used the technology that we now use in a lot of monoclonal antibodies to mean that it got more recycled. So instead of giving it monthly, you can give it once to cover yourself for 5 months, which is the length of an RSV season. And so we were involved with Terry and an international study looking at moderate preterm children, so between 29 to 35 weeks, and the efficacy over the first 5 months was against medically attended RSV lower respiratory tract infection was 73% and 80% against hospitalisation. And you can see here the numbers of children who, in the placebo group, who need to see a doctor or get hospitalised is significant in premature infants, because they lack the amount of antibody transfer that occurs in that last trimester. So this was very promising and led them to go on to a phase 3 study. But the other important aspect of this is how much is RSV responsible for all lower respiratory tract infections in that age group. And you can see here, if you're looking at hospitalisations, over 40% were prevented by the RSV vaccine. And we're getting increasingly aware of the importance of RSV in also giving rise to pneumonia, particularly pneumococcal pneumonia, during this sort of out-of-season RSV epidemics. This has become much more obvious and something I don't think we previously really were so aware of. And so this is the phase 3 study that was done with nirsevimab that looked at infants over 35 weeks and term infants. So looking at this is maybe this could be a universal approach, so not just giving it to high-risk groups, giving it to every infant. Clearly it will have to be costed as such. And the initial results that occurred in infants prior to the pandemic was published earlier this year in the New England Journal. And this is the Kaplan-Meier survival curve, and this is the point estimate, which is pretty similar to the preterm infants at over 70% for medically attended lower respiratory infections, a slightly lower estimate for hospitalisation, but you can see quite wide confidence intervals that overlap 0 but went up to 86. So again, this was promising. Now, more recently, the data was presented for the whole cohort, so that was 1,500 children that were enrolled before the pandemic. There was a cessation of enrolment and then it started up again, which is when we got involved in this study. And you can see very similar efficacy against medical attendance, hospitalisation, and very severe RSV requiring oxygen or intravenous therapy. And so this is a very highly effective antibody and much higher than palivizumab as a single dose over 5 months. So very pleasing efficacy studies. Now, the question that I sometimes get asked is, so when are we going to give it? Should we just give it to every baby when they get their hepatitis B vaccine, which would seem as one way of operationalising it? The problem is that if you're born in September, that means your antibody will be no longer circulating or at a very much lower level by May when the RSV season traditionally starts in Australia. And so it's clearly going to have to be delivered before the RSV season for children who are born from September to April. But it may be that during the RSV season, we might be giving it in hospitals with the hepatitis B, or maybe we'll be giving it to GPs a week later because children are being discharged from hospital so quickly. So I think this will have a significant impact on the amount of young children attending doctors, of 93 per 1,000. It works against both RSV A and B subtypes, impacts on overall hospitalisations for pneumonia, and it's been safe with no problems so to date with anti-drug antibody, which does occur, but it also is found in placebo recipients. It's likely to be licensed in Australia. We're hoping it will get submitted to the TGA later this year, so hopefully within 18 months we might have an approval. But how we use it is going to be determined by the cost and obviously the cost effectiveness. So unfortunately Australia, I think we have good data. But also there is another monoclonal RSV produced by Merck that appears to have similar high neutralising antibody levels that persist for 5 to 6 months, is well into phase 3 trials. So hopefully this will help drive the price to cost effectiveness. Maternal RSV vaccines. Terry mentioned the Novavax study, which started about 8 years ago and was very close to demonstrating efficacy, but didn't quite make it. And unfortunately, the point estimate, although overall prevention of medically severe lower respiratory infection was about 40%, the confidence interval overlapped with 0. But the real issue here was that actually in the US cohort, the efficacy was much lower. Probably as a result of the timing of the vaccination in relation to the RSV season, and most children, most mothers being vaccinated very late in their pregnancy. But one of the important factors was that you can see this impact on all-cause RTI, particularly driven for severe lower respiratory infections of almost 40%, partly driven by South African infants in particular, where the point estimate was even higher and represented about a third of the cohort. But certainly it was a proof of principle that this is a safe vaccine, this safe approach that will impact. This is for the first 3 months of life. So this led to 2 large maternal efficacy studies. As I said, the GSK study has been stopped for undisclosed safety effects that are now being looked at in more detail. But Pfizer had a similar size study that was enrolling during the pandemic, and recently, 2 months ago in November, its results were announced. The full study has not been published, but you can see here that the vaccine efficacy over 6 months, so 6 months post-vaccination, remains high, as it does for medically attended any medically attended lower respiratory infection. Data for hospitalisation is not yet been released. It's in publication, and we're hoping that it will have a similar rate of efficacy against hospitalisation. So this is very promising, and is likely to be submitted to the FDA this year. So what about older adults? Now, I just put these numbers up here to remind me that RSV is a big deal in older adults. I think in Australia, unlike children, we have not been so good at saying how much RSV or looking for it and monitoring it, which is going to make it more difficult when we want to introduce RSV vaccines. Novavax also had an unadjuvanted protein vaccine that was unable to show efficacy in older adults, but more recently there's been 3 large studies that have been undertaken by GSK, Pfizer and Moderna, who is not on this slide, with an RNA vaccine of 20 to 35,000 adults, and there are now 2 other companies with viral vector vaccines that are in phase 3 trials, and I'll show some of that data from the Janssen study. Now, the outcomes here are somewhat confusing because not every company has got the same outcomes, which is a shame. And so the 2 sort of endpoints that are looked at is medically attended RSV with at least 2 symptoms, of which for that is cough and sputum production are the most common ones, but could be shortness of breath or tachypnea, or 3 symptoms, which is a much more severe lower respiratory infection that includes shortness of breath and tachypnea, and more likely to require oxygen and hospitalisation. Unfortunately, neither of these studies are able to demonstrate an impact on hospitalisation because they're not large enough. And this is the results from the Pfizer study that was presented at a recent ACIP meeting. And again, you can see high levels of efficacy, even higher against the more severe disease, which are significant and certainly demonstrate an unadjuvanted bivalent vaccine can prevent RSV disease in adults. And there didn't seem to be a big difference in efficacy as you got older or if you had comorbidities. So very promising. The GSK, which is an adjuvanted vaccine, so it includes the AS01 adjuvant, including in Shingrix, is more reactogenic. The Pfizer vaccine, the reactogenicity was not dissimilar to the placebo. So you are seeing more local pain at the site of injection, but very rarely is it severe. And you are seeing a little bit more of fatigue, headaches, myalgias as a result of the adjuvant. But no serious adverse events that were attributed to the vaccine were documented. And again, a similar or perhaps an even higher point estimate, although quite wide confidence interval, so overlap with the Pfizer study, against the GSK, slightly different definitions, including all RSV-proven respiratory infections, lower respiratory tract infections with 2 symptoms and or a sign, or more severe RSV, lower respiratory tract infection, which essentially was increased numbers of signs and symptoms, or the investigator felt it was severe, and very high levels of efficacy. But the numbers are not great in these more severe definitions, so even though they've recruited 25,000 participants. Pleasingly, in these studies, they've also looked at persistence of antibody, and this is the GSK vaccine, and you can see that really the antibody levels remain much higher out to a year. So it may well be that this is a vaccine we don't need annually, but we need to be giving it every couple of years, or wouldn't it be great if it could give it every 5 years, but that data is not yet available, and it may not be possible with a viral respiratory infection. I think it's important to note that these studies were done over a time when the RSV epidemiology was slightly different, so maybe have an impact on point estimates, but certainly I think we've got evidence of highly effective vaccines. And if we look at all of them together, I won't go through this table. This has got the diagnoses and the point estimates of efficacy, but they're all pretty similar, including the Moderna, which has only just been released, the outcome of a very large study with similar efficacy estimates, with similar outcomes to the Pfizer, maybe a little bit better, but closer to the GSK or the more severe disease, they look pretty similar. So it's hard to know whether these different. This was a phase 2b study from Janssen, which again showed high levels of efficacy, similar to, and importantly, also appear to last even to the second RSV season. Although, again, these studies haven't been published, so it's a bit hard to drill down on this too much. So I think there's clearly an unmet need. We've got multiple vaccines that appear to work well, and all of them seem to work across the age groups. They can be co-administered with flu vaccines, but I think we need more studies in very at-risk populations such as immunocompromised adults. And we also need to be aware that we'll need to look out for rare side effects. So both those efficacy studies from GSK and Pfizer have had a case of Guillain-Barré syndrome in them. So that is something that we will need to be aware of and have the surveillance for acute flaccid paralysis in older populations as well as we do in children currently. And just finally, this is where we're at with paediatric vaccines. There was 2 adenoviral vector vaccines that unfortunately, I'm not sure are going to progress. The GSK vaccine has gone back to the preclinical stage. I'm not sure the Janssen vaccine is going to progress. I do think the impact of safety issues in adults is important for developing these vaccines for healthy children, even though we would expect not to have those same side effects. There's a live attenuated vaccine candidate that Sanofi is developing that's come out of Ruth Karen's lab that is interesting design, early studies, and Moderna is also starting studies looking at RSV and also combination RSV and human papillomavirus vaccines. So just finally, I think it's a real breakpoint time for RSV. I think we will have effective vaccines and monoclonal antibodies that we'll be able to use in children and at-risk adults. Maternal vaccination also looks as though this would be highly effective, and this might be a better option for low and middle income countries, and it's good to see they are being included in a lot of these efficacy studies, with a third of the children being in low middle income countries, particularly South Africa and Argentina. I think older adults have a lot to gain here, so Australia needs to beef up its surveillance, and I guess notification's a good start, but you've got to test. And finally, I think it's going to be a little while before we get RSV paediatric vaccines, but they look promising. Thank you to the people who've been involved in those studies, and I don't know all the answers, but I'm happy to take any questions.
Peter, thank you. So, Bill?
Peter, that was great. I just wondered about the mutability of the fusion protein. Is it thought to be fairly non-mutable, and so that's not going to be a problem for the vaccines?
So to date, there's certainly mutations that occur in the fusion protein, not to the degree that we see with COVID spike protein, obviously. To date, there's a paper coming out that we've been involved in a global surveillance for RSV isolates, looking at what's happened over the last 25 years, and to date, there's been no mutations that have affected the efficacy of the nirsevimab monoclonal antibody. But that is a risk, and so we will have to be doing that. And I think it's good that some of the sequencing for COVID is flowing over to sequencing for RSV, because I think that's actually teaching us about where these viruses are coming from and how they're spreading. So not a problem so far, but yes, need to watch.
Will the introduction of a no-fault vaccine policy, side effect policy, make a difference to the introduction of some of these new vaccines?
I thought we'd already introduced it, haven't we? No, that's just for COVID, isn't it? Yeah, look, I mean, absolutely. I think we need to have a process for providing compensation for people who do appear to have a vaccine injury. I think everyone, well, I think most people in the vaccine research field would agree, in the vaccine provision field. I think in government they need to be convinced, but I think the benefit to society would be significant, and I think it helps defuse the anti-vaccine movement, so I think that would be a good idea.
Ian?
Peter, the GSK failed maternal trial. Presumably the smoking gun is the adjuvant, and do you have any thoughts on that, or do you think they will go back with an unadjuvanted vaccine?
Yeah, sorry, I should have said it's a different vaccine to the elderly, so it's unadjuvanted in the maternal vaccine. It's an alum adjuvanted vaccine. So I think actually, no, yes, there's some things I cannot say being an investigator. So, but yeah, I don't think it's uncertain whether it's directly related to the vaccine or not at this stage.
Can I comment on that? Was not an investigator, was about to become an investigator, but we ended up, the study was prematurely closed because of this, just as we're about to enrol. It's actually in the public domain, Peter. So there's an excess of neonatal deaths in the vaccine group, and it's weird. They've done a lot of very detailed follow-up now. Does not appear to be related to any particular factor that might be identifiable. There are some risk factors in the way in which the groups were allocated that might explain it. It's sort of it's a real problem, because for me at least, my feeling is that something like this could potentially, particularly if it's unanswered, puts back doing pregnancy vaccine research big time, and especially if it's just not clear what the hell went on. But Frank, my own belief is it's probably nothing to do with the vaccine.
Yeah, and
but they've taken it very seriously, to their credit, and what they'll do long term with this in pregnancy, I don't know. They certainly have suspended it for the moment.
I think it was particularly prematurity was one of the risk factors, and particularly in low and middle income countries. So that's interesting. I wasn't aware that it had been publicly released, but I do think it's important to understand. And obviously what's happening is they're now following up the pregnancies that were already being vaccinated, and the effect perhaps looks less so. So I think it's uncertain that there may be no single. And this is always a problem when you try and have more people in a study by having a 2-to-1 randomisation. The opportunities for an imbalance of adverse events becomes higher. So I think that was also one of the contributing factors I think Terry was alluding to. That was the randomisation. So it's not adjuvanted, and I don't think we'd give an AS01 adjuvant to a pregnant mum, not at this stage. But, you know, mRNA vaccines we are, so we need to think about it, don't we, if they provide the benefit.
Peter, thank you. Perfectly discussed. Thank you.